The overall goal of this RNA transfection assay is to use HSP27-specific small interfering RNA to assess the potential role of HSP27 in corneal epithelial wound healing.
This method can help answer key questions regarding the function of His-Shock protein 27 during epithelial wound healing process. The main advantage of this technique is that small interfering RNA is a synthetic RNA duplex created to specifically target a messenger RNA transcript to induce its degradation, and it has been used to identify novel pathways in various cellular processes. Though this technique can provide insight into corneal epithelial wound healing, it may also apply to other systems such as skin and mucosa. Demonstrating the procedure will be Soon-Suk Kang, a technician from my laboratory.
To begin this procedure, culture human corneal epithelial cells in a 6-well plate with bronchial epithelium growth medium at 37 degrees Celsius and 5% carbon dioxide until they reach 95% confluence. Next, dilute 2.5 microliters or 7.5 microliters of the transfection reagent with 100 microliters of reduced serum medium for transfection. Then, dissolve HSP27-specific siRNA and scrambled control siRNA in 100 microliters of reduced serum medium to create 10 nanomolar of HSP27-specific siRNA and 50 nanomolar of scrambled control siRNA, respectively. Following that,